Hello students, welcome to class 11 biology. Today we will learn unit 3 cell biology and biomolecules. In this lesson we will learn about the cells and cell organelles. To learn about the cell, first we have to view them. View is seeing the cells. We can see the cells under the microscope. Robert Hooke was the first person to observe the honeycomb like structure in the cork, and that forms the basis for the cell. Okay, so finding of the cell. Okay, so in the first paragraph it is about the scientists who are describing each and every cell in a different way based on their observation. Robert Brown described the spherical body in the plant cell as nucleus, and Anton von Leeuwenhoek he said that the unicellular particles are the small animalcules. And then Sheldon, Matthias, and Theodore Schwann they gave the outline for the basic feature of the cell theory, and the Rudolf he explained that cell theory by adding the features stating that the living cells arise from pre-existing living cell. Okay, so discovery Aristotle was one who recognized animals and plants consist of the basic units. Okay, with this forming the cell. To observe the cell, these are all the here scientists who just propose the model and their views on the cell. Okay, so coming to this, microscopy is the interesting topic where you have to learn about the microscopy. Okay, so based on the resolution and based on the aperture and based on the facilities available in that microscope, we will be able to view different parts of the cell. Microscope is inevitable. Inevitable means it is unavoidable instrument in studying the cell subcellular structures. Okay, so here we have the microscope who has compound microscope was invented by Z. Janssen. So the microscope which we usually have in our laboratory, that is the compound microscope, it is basically works on the lens system and its properties of light and lens as reflection, magnification, and the numerical aperture. The common light microscope where the light is falling onto the mirror, which in turn is getting reflected to for us to view the objects. Okay, so the light microscope which has many lenses are called as compound microscope. So first it is getting magnified, furthermore it is getting magnified, that is what is called as the compound microscope. Bright field microscope is a routinely used microscope for the studying various aspects of the cell. So in this microscope, it allows the light to pass through the specimen and it shows well distinguished image. What is well distinguished? When you want to see the nucleus, it will be thicker. Okay, so because of the content from the different portion of the specimen, the contrast can be increased by staining the specimen. Staining of the specimen we have already learnt. Do you remember how we can stain the specimen by removing all the water and putting it into the alcohol, and you are putting the stain to make it colored. The light rays are focused on the condenser on the specimen on the microslide placed upon the adjustable platform, that is called as the stage. Light comes from the compact fluorescent lamp, that is CFL, and light emitting diode, then passes through two lens systems. Okay, so there are different magnifications by which the lens can be used, which can be rotated and fixed at the certain point to get the required magnification. So much ever you magnify, you will have the clear image. Much more magnification also, you will get blurred image. Okay, so the magnification to the optimum level will give the correct image. It works on the principle of the numerical aperture value and its own resolving power. The first magnification of the microscope is done by the objective lens, which is called as primary magnification, and it is a real and inverted image. The second magnification of the microscope is obtained through the eyepiece, that is called eyepiece lens. It is called as a secondary magnification. So the specimen is getting magnified one time, and again it is getting magnified because of the presence of another lens, so that we get a good magnified image of the proper resolution. Then electron microscope was first introduced by Ernst Ruska and developed by Binning and Roher. It is used to analyze the fine details of the cell organelles. Okay, so these are all the small organs that is present inside the cell. That is called as the ultrastructure of the cell. It uses beam of accelerated electrons as a source of illumination. Okay, so electrons are sent through the cell, okay, as a source of illumination, and therefore the resolving power is 1 lakh times greater than that of the light microscope. Okay, so when you want to see the clearer image of the organelles, you can use the electron microscope. The specimen to be viewed under the electron microscope is dehydrated. Dehydrated is a removal of water, and impregnated. Impregnated is kept on top of with the electron opaque chemicals like gold or palladium. This is essential for withstanding the electrons. Okay, we cannot keep it onto the normal slide. There are two types of electrons, that is transmission electron microscope, scanning electron microscope, that we will learn in detail now. What are the parts of the transmission electron microscope? Electron generating system, electron condenser, specimen objective, tube lens, and projector. So first the beam of electron is passed through the specimen to form the image on a fluorescent screen. Okay, so whichever you are viewing, you are not viewing the specimen, you are viewing it on the screen only. Do you follow? In the light microscope you will see the specimen as such, but in the electron microscope you will see it on the screen. The magnification is 1 to 3 lakhs times, and the resolving power is 2 to 10 angstrom, and it is used to study the detailed structure of the viruses also. They are very small structures. Mycoplasma is a kind of fungi and cellular organelles. Okay, that is about the transmission electron microscope. Scanning electron microscope, what is the major difference is that it is providing us with the three-dimensional image. You know the thickness also of the specimen. Okay, so on the lower resolving power, and it is having it than the transmission electron microscope. In this, electrons are focused by means of the lens into the very fine point. The interaction of the electrons with the specimens result in the release of the different forms of radiations. Okay, so from the surface of the specimen, these radiations are then captured by appropriate detector, amplified, then imaged on the fluorescent screen. Okay, so here you have the 2 lakhs times resolution, 5 to 20 nanometer in size, you can find with the help of the scanning electron microscope. So now we are getting into the actual cell theory. The German botanist Matthias Sheldon and Theodor Szechuan proposed plant and animal cells form the basic building blocks of the life. All the organisms are made up of cells. New cells are formed by the division of pre-existing cells. Cells contain the genetic material which is passed from parent to daughter cell. All metabolic reactions takes place inside the cell. So exception to theory, viruses are puzzles. So exception means this will not fall under those characteristic feature. Viruses are puzzles of biology because viruses are viroids, prions, or these definitions we have learnt in the previous lessons. Do you remember? Are the exceptions to the cell theory because they lack protoplasm. There is no protoplasm seen in the case of viruses, which is the essential part of the cell, and they exist as the obligate parasite. Obligate parasite. They can live as a parasite only when they are not there inside the cell. Once they are outside the cell, or the animal cell or the plant cell, it will become crystalline. Okay, so it becomes the stone like. Okay, so this is the exception for the cell theory. Coming to protoplasmic theory, here protoplasm, the term was given by Purkinje, and here he coined the term protoplasm for the sap inside the plant cell. What is sap? It is a mineral and water together. It is forming the cell sap. Usually it is found inside the vacuole. Okay, Hugo and Mohel indicated the importance of the protoplasm. So here they established the similarities between the protoplasm and the sarcode and proposed a theory which is later on called as protoplasm theory. Okay, initially they found the cell which was seen under the microscope, and we have learned about what is light microscope, what is electron microscope. Under electron microscope you have two types of electron microscopes, that is transmission electron microscope, scanning electron microscope. Later on we have studied what is cell theory and what are the characteristic features of this cell, and exception to cell theory is the virus, which is acting as a biological puzzle and it becomes stone like once they are out of the living cell. And then we have learned about what is protoplasm and what are the characteristics of the protoplasm. That is what we are going to study. It is a physical basis of life. Cell is a building block of life and it is a physical basis of life. Protoplasm is a physical basis of life. Protoplasm as a colloidal system. Protoplasm is a complex colloidal system which was suggested by Fisher in 1894 and Hardy in 1899. It is primarily made of water and various other solutes. What are solutes? Which is able to dissolve in water. They are called as the solutes of the biological importance like glucose, fatty acid, amino acid, minerals, vitamins, hormones, enzymes, etc. These solutes may be homogeneous. Homogeneous is soluble in water, it becomes a part of it. Heterogeneous mass, that is insoluble in water, it is able to we are able to distinguish them from the water. So that only gives the colloidal nature for the protoplasm. Physical properties of protoplasm, it is very important. The protoplasm exists in the semi-solid condition. Semi-solid, it is not solid, it is not a liquid. Okay, it is a gel condition due to suspended particles and various chemical bonds and maybe the liquid state. Okay, protoplasm which is in gel form can change into sol from solation, and also sol can change into gel, that is called as gelation. So it is also called as sol gel theory. Okay, sol gel theory, it becomes a solid like or it becomes a liquid like. Okay, so sol gel condition of the colloidal system are the primary basis or prime basis for the mechanical behavior of the cytoplasm. Protoplasm is translucent. What is translucent? It is not transparent or opaque. It is translucent in between. Odorless and is a polyphasic fluid. It is appearing in a different phases. That is liquid, solid. Okay. So it is a crystalline colloidal solution which is a mixture of chemical substances forming crystalloid, that is just true solution, and other forming the colloidal solution. The most important property of protoplasm by which exhibit three main phenomena, that is Brownian movement. You are familiar with the Brownian movement from chemistry, I think. So that is exhibited by colloid and amoeboid movement. It is a movement of the insoluble material like a amoeboid fashion, and cytoplasmic streaming. Cytoplasmic streaming or cyclosis, a movement of the cytoplasm. The viscosity is nothing but the thickness of the protoplasm is 2 to 20 centipoises. The reflective index of the protoplasm is 1.4. This is important, one more question. Protoplasm reflective index is 1.4. The pH of the protoplasm is around 6.8. It contains 90% of water. 34 elements are being present in protoplasm. Protoplasm is a neither good or bad conductor of electricity. And cohesiveness, so particles and the molecules in the protoplasm adhere with each other. Okay, they are just getting attached with each other. Okay, and by the force called as the van der Waals force. Okay, contractility of the protoplasm is important for the absorption and removal of water from the during the stomatal operation. Stomata, do you remember? It is a pore on the leaf. Okay, so how this pore is getting opened? By the accumulation of water. Accumulation of water only makes the stomata to open. That can be happening in the protoplasm. So why? Because it is able to accept the water and also to release the water. Surface tension. Protein and lipids of the protoplasm have less surface tension. Hence they found at the surface forming the membrane. Okay, on the other hand, the chemical substance sodium chloride have high surface tension. So they occur in deeper parts of the protoplasm. Okay, next coming to the next topic, that is cell size and shape. It is they are found in different sizes and different shapes. Okay, group of similar cells to form the tissues. Tissues forming the similar function form the organs forming the same function organ system. So these organ systems together forms the organism. Okay, the shape can vary from cocci. Cocci is rounded form or rod shaped, or it can develop the hexagonal shaped or T shaped. Okay, globular, elongated, spore, and cylindrical shape. There are different shapes and sizes of the cell. Okay, so types of cells. So this we have read in detail already about the prokaryotic cell, mesokaryotes and eukaryotes. So you know what is prokaryote, which is not having the true nucleus, then they are called as the prokaryote. A mesokaryote. So scientist George and his co-workers proposed another kind of organism, mesokaryote. These organisms, which share some of the characters of the prokaryote, some of the characters of the eukaryote, are called as the mesokaryotes. They are in between the prokaryote and eukaryote. These contain well organized nucleus and nuclear membrane. DNA is organized into chromosome but without histone protein. Histone protein is present in the case of the eukaryote. Okay, so then they divide through amitosis, similar as prokaryote, like binary fission. Okay, mitosis is not seen in the mesokaryotes. Eukaryotes undergo mitotic and meiotic cell division. Do you follow? So histone protein not present, amitosis occur, and certain protozoan like Noctiluca, some phytoplanktons like Gymnodinium, Peridinium, and dinoflagellates are the representatives of the mesokaryotes. Eukaryote, we have learned so many things about eukaryotes. They have the true nucleus, DNA associated with the protein, special protein called histone protein. Membrane bound organelles are present. Few organelles may arise by the endosymbiosis, and which is the cell living inside another cell. The organelles like mitochondria, chloroplasts are well supported by this theory. Okay, so this prokaryotes, eukaryotes, what are the differences among them? This is a very known and familiar points to you. This we have learnt in the first lesson also. Please read through. If you have done any doubt, you can definitely ask me. Okay, so when you are coming to the structure of the plant cell and animal cell, ultrastructure, when you say, it is about the internal structure. So coming to this animal cell, they are bounded by the cell membrane, the plasma membrane. Cell membrane is otherwise called as the plasma membrane. Inside this membrane, gelatinous matrix called protoplasm is seen, and they contain the nucleus and cell organelles. Cell organelles are a endoplasmic reticulum, mitochondria, Golgi body, centriole, lysosome, ribosome, and cytoskeleton. Here you have the presence of the plant cell. A typical plant cell has prominent cell wall, okay, clearly visible, and it has got the large vacuole and plastids are nothing but the chloroplast. So protoplasm it is a living content of the cell. Protoplasm is a living content of the cell, and it is surrounded by the plasma membrane. It is a colorless liquid material existing throughout the cell. Okay, so this cytoplasm and nucleus together only constitute the protoplasm, and it is composed of the small particles and then the liquid material. Okay, difference between plant cell and animal cell is given. Please read through. Okay, so the protoplasm appears like a colorless jelly and gelatinous viscous elastic granular material. It appears foamy due to the presence of the large number of vacuoles. It responds to the stimuli like heat, electric shock, chemicals, and so on. Okay, then coming to cell wall. What is cell wall? It is the outermost protective layer found in the plant cell. It is found in bacteria and fungi also. Okay, so whereas it is absent in the animal cell, it is actively growing portion, being it is a non-living material. It grows by the accumulation, okay, deposition of the polysaccharide. The cell wall can grow. It is growing up as a complex material in various organisms. In bacteria, it is composed of peptidoglycan. This is important. Bacteria cell wall is made up of peptidoglycan, fungi, chitin, fungal cellulose, and algal cellulose, galactans, and mannans. In plants, it is made up of cellulose, hemicellulose, pectin, lignin, cutin, suberin, and silica. Plant cell wall shows three distinct layers. This is important. So primary layer, secondary layer, and middle layer. It is described as primary wall, secondary wall, and middle lamella. Okay, so the primary layer is the first inner 2D. Middle lamella primarily consists of the loose network of the cellulose microfibril in the gel matrix. It is very thin, elastic, and extensible. It is able to stretch itself. In most plants, fibrils are made up of cellulose oriented differently based on the shape and thickness of the wall. The matrix of the primary wall is composed of hemicellulose, pectin, and the glycoprotein and water. Hemicellulose binds the microfibril with the matrix and glycoprotein. Okay, so they control the orientation of the microfibril, while pectin serves as a filling material of the matrix. Cells such as parenchyma, meristems have only primary wall. So here the secondary wall is laid during maturation of the cell. It plays a key role in determining the shape of the cell. The thick inelastic and is made up of cellulose and lignin. The secondary cell wall is divided into three sub-layers termed as S1, S2 and S3, where the cellulose microfibrils are compactly arranged with different orientation, forming a laminated structure, and cell wall strength is increased. Middle lamella, it is the outermost layer of the calcium, and it is made up of calcium and magnesium pectate deposited at the time of cytokinesis. It is a thin amorphous layer which cements two adjacent cells. It is optically inactive. Plasma desmata and pits. Plasma desmata act as a channel between the protoplasm of the adjacent cells through which many substances pass through. Moreover, at few regions, the secondary cell wall is laid unevenly, whereas the primary wall and middle lamella are laid continuously. Okay, so and such a where there is no continuity, they are called as the pit. Pit is adjacent to the cell and it is opposite to each other. Each pit has got the pit chamber and it is covered by the membrane called as the pit membrane. So pit membrane has got many minute pores. They are permeable. Okay, so they are permeable. It is allowing the materials to pass through. Functions of cell wall: it offers defined shape, rigidity to the cell. It serves as a barrier for several molecules and provides a protection to the internal protoplasm, prevents bursting of the cell due to osmotic pressure, play a major role in acting the mechanism of defense for the cell. The cell membrane is also called as the cell surface or plasma membrane. It is a thin structure which holds the cytoplasmic content is called as the cytosol. It is extremely thin. Okay, so that is what you are going to learn about the cell membrane. There is a model proposed that is called as the fluid mosaic model. It says that the plasma membrane is made up of lipids and protein, okay, with a little amount of the carbohydrate. Lipid membrane is made up of phospholipid. Phospholipid is hyperbolic, hydrophobic tail and a hydrophilic head. Okay, so the hydrophobic tail repels water. Hydrophobic phobia is always the it is not accepting. Okay, or it is not it is not going along with water. Okay, hydrophobic, it is water hatred. Okay, so hydrophobic tail and hydrophilic, which is able to dissolve in water. Hydrophobic tail repels water and hydrophilic head attracts water. The proteins of the membrane are globular protein. The protein is described as a globular protein which is found in the in between the lipid bilayer. Two layers of lipid and in between you have the presence of the protein. So in between the proteins are present, they are called as the integral protein. Few are superficially attached. On top you can see the protein, they are called as the peripheral protein. Follow? So we are learning about plasma membrane from previous paragraph. Plasma membrane is made up of lipid and protein. Lipid is made up of two part, hydrophobic head and hydrophobic tail and hydrophilic head. Okay, hydrophilic is water attracting and hydrophobic is water repelling, and then protein is present. Protein, if it is present inside in between these two layers, they are called as integral protein. If they are superficially on top, if it is present, they are called as the peripheral protein. And sometimes this carbohydrate is present, they are called as the glycocalyx or glycoprotein. Glyco with reference to the carbohydrate. The movement of the lipids from one side to another, that is by the flip-flopping movement or flip-flop movement. The movement takes place more slowly by the lateral diffusion of the lipid molecule. Phospholipids have the flip-flop movement because they have smaller polar regions, whereas proteins cannot flip-flop because of the polarity. Okay, so the function of the cell membrane, the functions of cell membrane is enormous. Okay, and it includes the cell signaling, transporting nutrients, water, preventing unwanted cell signaling, transporting nutrients and water, preventing unwanted substances entering into the cell, and so on. Next, coming to cytoplasm. So cytoplasm is a non-living material. Once the cytoplasm is added with nucleus, it is called as protoplasm. Cytoplasm plus nucleus only gives the protoplasm. Okay, so it is made up of all inorganic substances and it is present inside the plasma membrane. Okay, so we will learn about cytoplasm in the next class. Okay, so for your homework, you please draw this fluid mosaic model in your classwork. Mention the name of the lesson, write date, and you give the heading model of cell membrane fluid mosaic model, and you draw this diagram and label the parts in your classwork. Thank you, students.